Yeah, thank you. Thank you so much for the introduction, and also thank you for the invitation. And yeah, as Sarah is saying, today I'm gonna talk about PRSet, which is a pathway-based polygenic risk score analysis and software that we have recently published. So yeah, as most of you might know, polygenic scores have been used in the last few years to try to do some stratified medicine work. In some cases, this has been more successful, for example, for breast cancer. In other cases, it's not still there, but there is some work already done, for example, in psychiatric disorders. So most of these methods about polygenic scores are using the current polygenic paradigm to model genetic liability in complex disease, where we have a disease model where individuals go from low genetic risk to disease to a high genetic risk. And so when we calculate polygenic scores, what we are doing is aggregating genetic risk variants that are scattered across the genome. And so for some individuals, the disease risk might be small, like for example, only 2 risk variants, whereas for other individuals, the genetic risk will be much higher. And usually cases will be clustering towards the high risk side of the distribution in the disease model. But what this model is not taking into account is heterogeneity. So for example, if we focus in the high risk part of the spectrum, there might be some cases, for example, that are cases because of one root to disease, whereas other cases might be cases for a different reason. So what we are proposing is a pathway polygenic paradigm where, for example, some individuals have a high risk, but that's given by pathway A, for example, whereas other cases have also a high risk, but this is given by a different pathway, for example, pathway B. So how to calculate this pathway-specific polygenic risk scores? And in order to explain this, I'm gonna use a Manhattan plot that is hopefully familiar with many of you, the schizophrenia from a few years ago. So in this Manhattan, we can see that we have all the genetic signals. And what we are proposing is that maybe some of them are given or are due to, for example, synaptic pruning in schizophrenia. And there are other genes, for example, that are due to immune activation. So what I am representing here is the blue boxes or the purple boxes are genes. And then the lines are the genes that are forming a pathway. They are not exons and introns. And then, for example, these other signals in green, they might be due to a cannabis consumption pathway. And importantly, in PRSet, these pathways are defined by the user. So the user defines what are the genes that are composing a pathway. And so the user can define as many pathways as he or she wants. And therefore, one single individual will have, instead of one single genome-wide polygenic score, they will have as many polygenic scores as pathways have been defined. So PRSet is the software that we have developed to calculate these pathway-specific polygenic scores. It was mainly developed by my colleague Sam, who is now working at Regeneron, and is an extension of PRECISE, which is a clamping and p-value thresholding method. So I like to present this slide because it gives a comparison of PRECISE, which is the genome-wide method for clamping and thresholding that we usually use, and PRSet. So for example, in PRECISE, the input is a GWAS summary statistics with the individual level, genotype and phenotype data. And PRSet will give us an input the same, but then we also need to provide the pathway information, which can be vet files or a list of SNPs or GNT and GTF files. Then for clamping and thresholding, PRECISE does it for a given R-squared and linkage disequilibrium window. PRSet does the same, but it will do that for each pathway separately. And then the polygenic score calculation, and in PRECISE, it will be one genome-wide PRS, whereas for PRSet, there will be many polygenic scores for K pathways. And then the results in genome-wide PRS, we will have the phenotype with regression on the polygenic scores. In PRSet, we will have the same, but then for an individual pathway. And also we have enrichment of GWAS signal in the pathway via permutation. And this enrichment is given to take into account how big the pathways are. So what we have in the paper are like two main parts. And I am gonna present these two parts also separately here. So the PRSet paper part one is a bit more focused on this type of results, the enrichment results of GWAS signal. Whereas the second part is a bit more, is using both results, the association and also the enrichment. So first I will present part one, then I will stop. And then if you all are, you're free to ask questions about that part, and then we can continue to the second part. So in the first part of the paper, what we wanted to do was to benchmark the power of pathway PRSs to capture GWAS signal. And in order to do that, we were comparing PRSet with two well-established pathway enrichment methods, such as MAGMA and partition LD score regression. So they use different ways to assess enrichment. We explained that a little bit more in detail in the paper, but at the end of the day, the three methods are giving us competitive p-values, testing for enrichment of GWAS in each individual pathway. And we benchmark it using different approaches. So at the beginning, or first, we evaluate the pathway enrichment using canonical pathways. And we start doing that by simulation analysis. So in these simulations, we are starting with as many pathways as we could find publicly available. So we were taking over 4,000 pathways from databases like Biocarta, Genontology, Reactome, GET. We were taking all the pathways there, and then we were selecting 50 random pathways and defining them as causal. Out of those 50 causal pathways, we were selecting SNPs, like between 1-30% of SNPs from each pathway, and then we will define those SNPs as the causal SNPs. We will get that list, and then we will calculate what is, for all the 4,000 pathways, what was their enrichment in causal SNPs. So by doing this, we will have a rank of pathways based on enrichment, and we will know what it is. We will know the truth. And then for all those pathways, we were also calculating the pathway enrichment using PRSet, MAGMA, LD score regression. And then we were comparing the GWAS pathway enrichment results from the three methods with the simulated truth, the rank of the pathways based on enrichment that we had calculated before. And these are the results for the simulations. What you can see in the x-axis are two different heritabilities that we simulated, 10%, 50%. And then in the y-axis, you can see the correlation between the rank of the pathways and the rank of the software for the enrichment. So I am presenting here the results only for a specific GWAS sample size, 125,000 individuals. And then we are separating the target sample size, and we are doing it for 1,000 individuals target, 10,000 individuals, 100 individuals. So this is the number of individuals for whom we calculate the polygenic scores. And you can see in red is PRSet, blue, MAGMA, green, LD score regression, that for 1,000 individuals, PRSet is not doing very well compared to the other two methods. But as the target sample size increases, PRSet does a little bit better. And when the target sample size is huge, PRSet becomes the best performing method. We observe the same results when we use different sample sizes for the GWAS. So the pattern is consistent when we use 50,000 individuals for the GWAS or 250 for the GWAS. So the main contributor here for the performance is the target sample size. So as take-home message, the target sample sizes for PRSet need to be around 10,000 people if we want some well-powered PRSet analysis. Then we use a different approach, and we were comparing the pathway enrichment using Malakar's scores. And for those who are not familiar with Malakar's, this is a database that provides a score for each gene and the contribution of that gene to disease. So we were taking the Malakar's database and taking the relevance scores for all the genes for a bunch of diseases that we selected. And then we were ranking those genes, like what is the gene that is the most associated with disease and which is the gene that is the least associated. And we were then grouping all these gene scores into pathways. So we will end up with a Malakar's score for each pathway from the 4,000 pathways that we have been used before. So now, instead of having a simulated truth, we have the rank of pathways based on Malakar's scores. And we were doing the same. We had the Malakar's ranks, and we were comparing that with the results from PRSet, from MAGMA, and from LD score regression. And these are the results here. What you see in the x-axis are the different databases that we were using to get the pathways and calculate the pathway Malakar's scores. So MP is for the mouse project, Gene Ontology, Reactome, KEGG, BioCarta. And then in the y-axis, we again have the correlation between the rank from the Malakar's, what was the most relevant pathway for a specific disease, and the results from PRSet, MAGMA, LD score regression. So here PRSet is in red, MAGMA in blue, LD score in green. And I think that here it's important to see that the results change depending on the database that we use to define the pathways, and also they depend on the disorder or disease that we are looking at. And then we also use a different approach that is completely different. We don't use here canonical pathways, but we were evaluating pathway enrichment using tissue and cell type expression specificity. And this was inspired, and basically we copied the approach from Nathan Skin, where they were dividing the expression of genes by their specificity in a given tissue or cell type. So here, what we define as a pathway or as a gene set is only, for example, the 10 decile there, which is the genes that are the most specifically expressed in a tissue or cell type. And that will be our pathway. So for a list of tissues and cell types, we were calculating the specificity. We were taking the top decile genes, and we were defining that as a pathway. And then we were evaluating that was the enrichment with, again, PRSet, MAGMA, LD score regression. I'm showing here the pathway enrichment results for schizophrenia. And what you can hopefully see is that we have results as expected. So for brain tissues, the three methods have a higher enrichment than for non-brain tissues. This using GTEx. The brain is enriched in GWAS signal for the three methods. Then we were also using Alzheimer's disease for different cell types, using the genes that were most specifically expressed in cell types. And we can see that microglia, for example, is the one that is picked up by PRSet as with the highest GWAS enrichment, which is reassuring. I must say also that here depending on the thresholds, we were getting different results. So this is something also to take a little bit into account. But the most important thing I think to keep in mind about this part of the paper is that PRSet is not a pathway enrichment method. Like, we are using it here as a pathway enrichment tool to assess how well PRSet is doing and whether we can then use PRSet for different purposes. But we were using all this only to benchmark how well PRSet is doing, and we were benchmarking that against two well-established pathway methods. So the conclusion of this part one is that for target sample sizes of more than 10,000 individuals, pathway polygenic scores have a similar power for evaluating pathway enrichment as MAGMA and LD score regression. But then PRSet has the distinct advantage that provides individual level estimates of genetic risk for each pathway. And this is something that MAGMA and LD score don't do. And it's also something that we can exploit for applications that are usually used in polygenic score approaches. So I'm gonna stop here. If you have questions about this part, now is the time, and then I can continue with the second part of the paper, which is a little bit different.
Great. Yeah. So if anyone has questions, feel free to raise your hands, type in the chat, come on video and just ask. Maybe I can start with this is just an observation. So when you showed the Alzheimer's disease enrichments for these cell types in the brain, LD score regression didn't seem to point to microglia, and that's a bit unusual because based on previous analysis, that's what has come out of LD score regression.
Yeah, we were wondering the same thing all the time, and that's why I made this remark, and we also put that comment in the paper that for this approach of defining genes based on specificity, it depends a lot, like the results were depending on whether we were defining 10 deciles or 20 deciles of specificity. And they were also depending on how we were putting the thresholds. So we are showing that result, but then we were also testing not only the top 10 decile, but then the trend towards the specificity. So because we observed that result, we were trying different approaches to benchmark it using different ways. We also used two different versions of MAGMA and benchmark it against that. So yeah, we saw that this specificity expression was a little bit flickering. This is something that I am working now to refine that part of it.
Yeah,
it's a good point.
And then maybe my second question was another one where on these, oh God, what was it called? These correlations of the different pathway enrichment methods with the, what was it called? Card something.
Cards,
yeah. The BMI was so strikingly different between, you had this positive correlation for the PRSet and then the striking strong negative correlation for one of the other two. Do you have any idea how that happened?
Yeah, well, the pathway databases are using different sources to define pathways. So then, yeah, we are presenting that broken down by all the different pathway databases because we were seeing these differences in the results in terms of the correlation and the significance. This is something that I'm gonna touch on later, and is that the pathways are defined not perfectly, right? And this is one of the limitations of PRSet and also the pathway enrichment methods, that depending how pathways and gene sets are defined, then the result will be dependent on that. And that's an important thing to keep in mind when doing all these analyses.
Very good. Does anyone else have any questions for this first part of the talk? If not, then maybe we move on. And of course, you can all ask questions about everything at the end of the talk.
Thank you. Okay, so in the second part of the paper, what we were doing was to assess the performance of pathway PRSs for disease stratification. And this is exploiting this feature of pathway PRSs that we can have individual level estimates of genetic risk. And this is something that is different from MAGMA, LD score, the other enrichment methods. So here, keep in mind that all the applications that have been used for genome-wide PRSs can be also used for the pathway PRS methods. So here, when we were thinking about how to do this, we were thinking about doing this disease stratification task in the what we think might be the most likely situation for doing this, which is that the GWAS that we have to calculate the polygenic scores is a case-controlled GWAS. As in, if we want to separate subtypes, we are not going to have a subtype GWAS. We won't have a case GWAS, or if we have it, the sample size won't be so huge as the case control. So we are starting with a case control GWAS and the betas that are from that GWAS. And then we will have a sample where we have subtype information at the individual level. And maybe that will be because we have a little cohort, for example, that has gone through a very deep phenotyping, and we have the subtype information for those individuals. So what we will do is to calculate pathway polygenic scores for that cohort for which we know the subtypes, and we will use it as a training sample. And that training step will have two main parts. The first part is going to be selecting the most important pathways. And we are doing that based on the pathway enrichment that I was showing before. So we will select only, for example, if we have 4 pathways, 4 pathway polygenic scores, we will be selecting those that are enriched in GWAS signal and the ones that I'm highlighting here in green. Then with those 2 pathways, we will put them together in a lasso regression. And the regression will be, the phenotype that we use for that regression will be the 2 different subtypes. And what the lasso regression is going to do here is 2 things. It will take into account the overlap between pathways, and also it will reweight a little bit the pathway polygenic score effect sizes, because now we are regressing on the 2 subtypes. We are not using now the case control information. So out of this training sample, we will have a model that is taking into account the information of enriched pathways and the subtypes. So it's like optimizing it for differentiating 2 different subtypes. And then with a test sample for which we don't know the subtype information, we will be assessing the model performance. This is what we do with the pathway PRS. For the genome-wide, we do the same. We use also a training and a test sample. For the genome-wide, we will select the best genome-wide polygenic score associated with the 2 subtypes. We will take what are the best parameters for that, and then we will apply them in the test sample as well. And then we compare the performance between the 2 approaches, the pathway polygenic score and the genome-wide. So here we were comparing PRSet with LassoSum, that is a pretty well-performing genome-wide polygenic score method, and with PRECISE. And because I was showing before that we need large target sample sizes for PRSet to be well-powered, but then we don't have individual level genotype and phenotype information for huge subtypes. So what we came up here is with creating pseudo subtypes of major diseases that were available in the UK Biobank. So for example, we were combining individuals with hypercholesterolemia and type 2 diabetes. And what we were doing was to meta-analyze the 2 traits, GWAS. So we will have a big meta-analysis GWAS that is taking into account 2 different diseases. And then as the subtypes, we will have trait 1, for example, HC, hypercholesterolemia, versus type 2 diabetes. And then we will try to separate the 2 diseases as if they were 2 different subtypes. And we were doing that for different traits like obesity, coronary artery disease, and we were like doing these pair comparisons. One thing also here so in the x-axis, you see the 2 comparisons that we were treating as subtypes, and then the performance of the different methods. So one thing to keep in mind is that with this approach, with PRSet, we have many different pathway polygenic scores, and we have a lot of flexibility in the model to improve. But for LassoSum and PRECISE, we only have 1 genome-wide polygenic score in our model. So there is not so much room for improvement. So what we did was to develop this, we did this preset shift method where we do the same as with preset, but here the gene boundaries are shifted 500 megabases. So that means that we are keeping the same number of pathways, the same number of genes within the pathways, everything, all the statistical approach is the same, but we are like missing all the pathway and the gene signal. All the biological information is gone because we are shifting the gene coordinates. And what you can hopefully see is that preset was doing quite better compared to the genome-wide methods. And also it was doing better compared to this preset shift where we are using also many different parameters to optimize the model. So this was quite encouraging. But then we wanted to do a bit of a more difficult task. And we were defining subtypes in a different way. So now the way that we were defining subtypes was by doing comorbid diseases. So the GWAS here to calculate the polygenic scores will be one single trait or disease. And then the subtypes will be defined as other traits that are comorbid but for that disease. So for example, our disease will be type 2 diabetes. And then the two different subtypes will be individuals with type 2 diabetes that have obesity. That will be subtype 1. And subtype 2 will be individuals with type 2 diabetes without obesity. And then we will try to separate the two of them. So again, I'm showing here preset shift, and the genome-wide polygenic score methods, lasso sum, and precise. And you can see that in most of the cases, the performance is much lower because also the sample size is much lower for these subtypes. But still preset was doing better than the genome-wide polygenic score methods. So we tried to complicate a little bit more this task, and we tried to use real subtypes. So here we were trying to separate the two well-established subtypes of inflammatory bowel disease. So we were trying to separate Crohn's disease versus ulcerative colitis. And we were using the IBD GWAS case control to calculate the polygenic scores. So here the performance, you can see the R score is really, really small for all the methods. Preset seems to be doing better, but is too small. But also note that the sample size here is 2,000, 3,000 individuals. And as I was showing in the simulations, we need much larger sample sizes for this method to do best and to be well powered. So then what we tried to do was also to use this approach to try to differentiate bipolar disorder. So this is a bit different because here what we had was the PGC, the Psychiatric Genomics Consortium data. So we had the genome-wide summary statistics for 55 different cohorts. So what we were doing was to split those cohorts. Some of them will be the GWAS. So I was taking 34 bipolar disorder case control cohorts and use them as a GWAS to calculate the polygenic scores. And then for the subtypes for our target sample, I was using 21 remaining cohorts. So my training data set will be 20 cohorts, and then I will be testing with one. And I was doing that iteratively. And in this case, it's important to see that even if the sample sizes, they are much larger than for IBD. So for bipolar disorder 1, we have almost 11,000 individuals. And for bipolar disorder 2, we have 3,500. So what you can see here is, again, that preset shift are doing better than the genome-wide methods. Preset is doing better than preset shift. And because we were using all these different cohorts, then the performance was a little bit different depending on the validation, the test cohort sample size. So that is what I'm representing here with the size of the different dots. So as a conclusion of this part 2 of the preset paper is that these pathway polygenic scores outperform the genome-wide methods for the classification of subtypes. And then we were hypothesizing why. In the paper, we have some supplementary notes explaining these things in more detail. So we think that in this case, the pathway polygenic scores are doing better than the genome-wide because we are taking here variants that are in genic regions. We are selecting relevant biological pathways. And that's what we see when we are comparing PRSet versus PRSet shift, the method where we were shifting the gene coordinates. But also we think that we are gaining performance, we are improving the performance because we have much more modeling flexibility, because we have a higher number of pathways that can optimize the prediction of different subtypes compared to the genome-wide methods. And then we explain this in a bit more detail in the supplementary note 3. So some limitations, future directions, thoughts that I came up with. Well, as I was commenting with Sarah, right, like pathway polygenic scores rely on pathways, but these pathways are not always well defined. So what we are thinking now is to use tissue, cell type, co-expression networks, like integrate more functional genomics data to improve how pathways are defined, and then apply them on pathway polygenic scores. Also, I didn't say this before, but here we are using genes, right? And the genes in pathways. So the SNPs that we are including in our pathway polygenic scores are SNPs that are within gene boundaries or around that, like 35, 10. The user can select how they want to expand the window around a gene. But we think that the pathway polygenic score method will improve if we can use and integrate it with SNP to gene mapping strategies. So a SNP that is far away from a gene but is implicated in that gene function is also included in the pathway polygenic score. Also, as you have seen in the second part, we are optimizing the method, like we are taking the pathways that are the most enriched from publicly available databases. But then we are putting them, all those pathways together in the lasso regression. So it seems that it is unlikely that for complex diseases, there is a silver bullet pathway that can stratify patients. We are doing much better when we are aggregating relevant pathways and then we are optimizing these pathways. But then the limitation here, or the bad thing about it, is that by aggregating these pathways, for example, with the lasso model that I was presenting before, this complicates the experimental follow-up because we cannot take one single pathway that we think that will stratify and will solve this task. And with this, I guess I spoke too fast because I did this in 36 minutes. But yeah, thank you for listening. If you have any questions about anything, this part or the second part, please do. And yeah, thank you to Paul, Sam, and everyone in the team.